Hello, everyone, and welcome to this audio podcast. This audio podcast is part of a Calyx report, the Calyx report edition. This audio podcast is part of a Bostix SandZtorm, and this audio podcast is part of a Squadron Communique, and this audio podcast is part of a Storm Bostix production. In this audio podcast, there will be a sort of an overview looking at animal models in context of uh, of uh, of nephrotoxic nephritis and glomerulonephritis in and from works from in around works from 2014, sort of peer reviewed and published works from 2014. So animal model or models are used during the are used during sort of research and investigation of human diseases. The animals selected will usually meet determined taxonomic equivalency to humans in order to react to disease or its treatment in a way that resembles human physiology. Research relating to nephrotoxic nephritis and glomerulonephritis can be sort of traced back to the 1960s and late 1970s, when observations were reported on the role of complement in nephrotoxic nephritis produced by rat models, or by a rat by sorry in the rat or in a rat model by either rabbit or duck nephrotoxic sera, or NTS. And in addition, the course of complement reactions in glomerulus in nephrotoxic nephritis is used as a measure of the course of immunological events in glomerulonephritis, or GN. The observations gave the first indication of of the duration of sort of the duration of sort of the immunological reactions, the reactions in a chronic form of this nephritis. And bear in mind again, viewers and listeners, that there are varying forms of nephritis, varying forms of nephritis, and linked to varying forms of renal disease. So, for example, systemic lupus erythematosus is a form of nephritis, or autoimmune sort of renal disease, as well as again sort of glomerulonephritis, and amongst others. And so and so and so finally, the differences among the roles of played by complement in nephrotoxic nephritis, NTN, produced by various rabbit and duck and nephrotoxic sera, suggest the possibility of different mechanisms of mediation of injury caused by these sera. Matrix metalloproteinase 9, or MMP9, is a conditionally expressed enzyme, and is upregulated in glomerulonephritis, or GN. And the researchers showed that leukocyte-derived MMP9 mediated the recruitment of pro-inflammatory macrophages in the kidney during experimental work in experimental research. And in the same works, the induction of nephrotoxic nephritis in wild type mice, or laboratory mice, resulted wild type mice in wild type mice in the laboratory environment, resulted in upregulation of MMP9, followed by leukocyte infiltration, albuminuria, and subsequent glomerulonephritis. So animal models of GN closely replicate features of human GN, or glomerulonephritis, and have provided insights into the disease. The reproducibility and uniformity of such animal models has also permitted a step by step dissection of many of the mechanisms involved. Nephrotoxic nephritis is produced experimentally by the intravenous injection of antibody directed against glomerular basement membrane, or GBM antigens of the recipient species. Most of the accumulated inflammatory cells in many forms of human and experimental GN or glomerulonephritis are macrophages. BALB-C murine models or mice models have been helpful in understanding pathogenesis in a number of renal diseases. In retrospective research, six-week-old female BALB-C murine laboratory mice models were induced with injections of 50 microgram or zero 0.2 milliliter of bacterial lipopolysaccharides or LPS from Salmonella Minnesota, or RE595, twice a week for 5 weeks by the intraperitoneal route. In all the murine models and laboratory models, given injections, features of polyclonal or B cell activation (PBA) and GN glomerulonephritis developed. This animal model presents features that often encountered in human glomerulonephritis, particularly those associated with bacterial infection, as well as autoimmune diseases such as systemic lupus erythematosus, or SLE. In contrast to commonly used animal models of immune complex mediated nephrotoxic nephritis, NTN or GN glomerulonephritis, BALB-C murine models murine laboratory models have been highly reproducible and require only short-term injections of lipopolysaccharide. This BALB-C and nephrotoxic nephritis murine or laboratory mice models still hold experimental validity in the investigations of glomerulonephritis in humans. In addition, the BALB-C murine models can still be helpful in the investigation of the GN glomerulonephritis autoimmune phenomena as well. The reproducibility and uniformity of such animal models has permitted a sort of a step-by-step dissection of many of the mechanisms involved. The WKY rat strain is also highly susceptible to experimental models of the sort of crescentic glomerulonephritis, or the CRGN, including nephrotoxic nephritis and experimental autoimmune glomerulonephritis, EAG. And these rats or the rat laboratory models had shown the development of NTN nephrotoxic nephritis phenotypes, including glomerular crescents, and more recently, more recent works, the crescentic glomerulonephritis had been induced on a normally resistant rat genetic background, and identifies Lewis strain on two genetic loci, CRG1 and CRGN1 and CRGN2, as a new potentially valuable model for nephrotoxic nephritis GN. The nephrotoxic serum nephritis NTS WKY rat has also been used in research. Macrophages and T lymphocytes are the dominant cell types present, and the intensity of cellular accumulation, particularly with the interstitium rather than the glomerulus has been found to correlate with the clinical and pathological severity of disease in animal models and in humans. That draws this audio podcast to a close. This audio podcast is part of the Calyx report edition. This audio podcast as part of a Bostix SandZtorm. This audio podcast as part of a Squadron Communique. And this audio podcast as part of a Storm Bostix production.